meters. The top at Salmon Bank, about 20 miles east of us, that seamount gets to within 60 meters of the surface, and it is a place where fishing takes place. So yeah, that's very beautiful coral in our foreground. That's Iridogorgia, and that's Iridogorgia bella. And reference maybe for, the beautiful shape, very, very conspicuous, and beautiful. And again, this is another one of this. I talked about these adaptations to position oneself in the dominant flow regime to feed. Again, that top portion where all the polyps are, their feed, they can reorient themselves depending on where the current comes from. And then you also see that kind of spiral shape. So water is actually like funneled through to the middle of that plume, so that way it allows it to feed more efficiently.
I'll tilt down to that center.
And a couple of notes about that fishing line. So that's something that we see in shallow waters quite a lot up here in the northwestern Hawaiian Islands. There's a lot of marine debris and other fishing gear that is basically because of the way currents flow through the North Pacific. It aggregates in the waters just north of us, and then the trade wind brings it to the shores. And so a lot of the shallow reefs and emergent land features on the beaches, you see a lot of marine debris. They're coming ashore regularly. And in response to this, NOAA and a lot of other agencies have done a great job. And since the early 1990s, coming back to the northwestern Hawaiian Islands, every year they have annual expeditions where partners from a lot of different agencies come, and they remove the debris from the shallow waters. And
there we go. Co-pilot
on that marine debris, you can see that there was a couple of barnacles. There was a pellet or gooseneck barnacles on that, and that's also interesting, and that some of this lost fishing gear or derelict fishing gear actually becomes a habitat for some things. And but it also is, of course, very detrimental by wrapping around organisms. In some cases, this fishing gear continues to catch fish even though it's not tended anymore, and that's what's called ghost fishing. And of course, a big impact in the world. And there's estimates that ghost fishing might account for about 1/4 of all fish that's caught, and then of course that's a bad thing if no one's tending it.
Okay, let's go on. As I try to follow him, I blow him around and then have to follow him more. A vicious cycle.
Hi Daniel, this is Andrea. There may be a shrimp in the center of that Iridogorgia. I'm not sure if you guys caught that.
Yeah. I think it's a little bit far from us, Andrea. We're
right now the cliff's about 20 meters.
But I did note it in my records. Thank you, Andrea.
And we should recenter back to like 045 or so.
Looking at the sonar right now, it looks more towards the north.
Okay.
So if you want to go up and push ahead and see.
I'm seeing everything's to my right now.
Yeah, I'm just looking at my sonar.
Okay.
So yeah, that's also a nice view. As you can see, right along that ridge line, you have a lot of these corals right up, all lined up on that ridge line, and kind of almost perfectly lining that ridge line. And that's because a lot of times, so these ridge lines are barriers to flow. So if the currents go, in this case, from right to left or left to right, they would have to move around this object, around this ridge line,
cool.
And as a result, that you would have higher current velocities right around these ridges. And yeah, that's of course the environment that some of these suspension feeders like corals and sponges like. So they it gives you kind of an indicator where you see corals or places where you might have better or more favorable current flow for feeding.
More of a kind of a cliff.
And there we have another shrimp
this.
there.
Yeah, we could do the next ship move.
Okay. What would you like the heading to be? Do you want to go up the ridge, or do you want to go the same way Sirius is looking right now?
Go my direction.
Okay, video.
because
Zoom on
look
the
at
shrimp.
his sonar right now too.
Yeah, I see that.
But the serial sonar is
Okay,
up higher,
hold there.
so you want to go more north.
Okay, so maybe 4.
Is that a mysid?
4.
Okay, you can keep
Yes.
zooming.
Yeah.
Copy that.
Just try to hold him.
Looks like a mysid shrimp. Yeah, and Scott
Fish,
agrees.
yeah.
So those are the opossum shrimps. So they have this pouch.
Would like to put in another ship move of 30 meters at 004
Turn into
degrees
follow.
at 0.2 knots.
Nice, beautiful imagery of this. Yeah, mysid shrimp. Yeah, commonly known as opossum shrimps because they do have this kind of pouch on their
Good
belly. And yeah, that pouch, they have carry embryos in there, just like in a marsupial.
Okay, should we let him drift off video? Okay. There he goes. Okay, and wide. Thank you.
Nice.
So what was our direction? 010?
Yeah, so Scott, pointed out a couple of things that was full of embryos. So you know that is a female, and a pregnant female. Depth right now is 1,160 meters. That's right as we're coming up on top of this local high.
That's okay.
Oxygen levels are now at right around 1 milligram per liter.
Feel free to drop
And
any targets
that
you
should
want,
be
though.
continuing to drop as we
Good
move
if
up
ever
this
come back
ridge.
here, or just makes it more
Again,
interesting.
the target
Yeah, export it.
we're aiming towards is get to the top of this ridge, which is at 700 meters. We do see a kind of very slight current flow here. Very, very net slide, so that's coming from west to east, but
Just gotta try something with the lights for a second.
Okay.
So that's with the lower swing arms off. Back on.
Doesn't make much of a difference. Huh?
No. Let me see the uppers. The uppers will make a big difference.
So, pilot,
Yeah.
can I request that when we start moving up this slope a little bit, that we kind of image some of these rocks up in the upper left-hand corner, as well as this weird tumulus structure that seems to be running right down the middle?
That's what I was kind of coming to center up on, was this...
Yeah, this is really cool. So what it appears to be is this almost like long tumulus structure of a pillow lava flow, as well as the pillow lavas that seem to have been preserved on the left-hand side of the view right now. But yeah, wow, this is just amazing.
Wow.
There it is, going off into the distance.
yeah, it looks almost like a pipeline.
Yeah. So what could have happened, I guess, here is that due to the slope of this surface, it caused whatever magma or lava supply that was providing this eruption just enough to just jet right down this slope. Could have been a lava tube type of source where the internal structure was actually hollow.
Hold there.
Wow, that is just so cool.
And I'll sidle to the port to look at a...
Yeah, very
rocks over
bizarre.
A really interesting question that geologists would be asking is, what is the age difference
Cliffish.
between that surface that it erupted on and the actual eruption that generated that really long tube. Oh my goodness, that's just so neat.
Can we brighten up Sirius to see if we can see more of that tubular shape from Sirius?
And to the left there is another primnoid coral and a Furriate sponge. That's another one of this Furria Neuroca erecta. And there's also an ophiuroid on top of that primnoid coral. And
okay. Alright, we can go wide on D2. And I will continue up.
Do you feel like there's a current pushing you? Or do you feel like there's no current right now?
Doesn't feel like much. I'm off the stick now. I'm going slowly to starboard.
Okay. Yeah, I'm just looking at the tether loop, and it's basically just straight out.
maybe if a quick snap zoom on that sea star.
Of course. Let me set down.
Ship is at target.
Excellent.
Yeah, you can get a nice view of that tubular structure from Sirius now.
Okay, video. We can zoom in and get lasers.
Yeah, that's a great view from Sirius.
Okay, tilting down.
I'm going to try and measure this thing out as we go up, see how long it remains the same exact structure.
That's all you.
Yeah, so that looks like another Goniasterid.
Great. I'll tilt down a touch. I want to come out just a hair to get all his feet. That's better.
Yeah, beautiful image here.
Oh, slipping.
All right, I'll let you get back to the.
Thank you.
Go ahead.
Okay. There we go.
Oh, it looks like it's got some odd yellowish or orangish patches in a couple of areas between the arms. What are those? Parasites or just color, natural color pattern?
I think it might be a scale worm on there. It's hard to say. Yeah, it looks like a pollinoid, but yeah, my best guess is yeah. Is that Chris?
Hey, Dan.
Hey, Chris.
This is Chris Ma.
Welcome back.
I was just about to point out the scale worms on the surface, but I think someone beat me to it.
Is that a common association or not really? Like, I haven't really seen that as much.
Not that I know of. This is a first as far as I know. Lots of scale worms live in association with sea stars, but in the deep, there aren't really a lot of the relationships are not a lot of the relationships are known. Most of the known occurrences are in shallow species. Plus, this one, this particular sea star, it actually looks like something that I'm working on from sort of the Gorda Ridge region right now. So when I say Mediaster, that's sort of a cover name until I have the new species description published later this year. But it is interesting to note because it's certainly a new relationship that I've seen, and it definitely looks kind of interesting because Hyposteria, that thing that we saw last time, had scale worms living on it as well. So it could be just that any large, sea star provides habitat for these things. Sea stars often have chemicals in the body wall that keep fish from eating them. So maybe they're just good natural substrate.
Yeah, that's very interesting. And again, highlighting the value of this kind of videos that not only gives you an indication of what species we might be looking at, but then it gives you a little bit about their ecology, what other species might be using it as habitat, what they might be eating. We've imaged several sea stars on their food source. So yeah, extremely valuable, not just in the study of taxonomy, but yeah, it gives you first glimpses at behavior of some of these animals.
This looks like the end of that tube.
Wonderful, thank you. I'll be watching.
Thanks, thanks, Chris. And now we can get back to that pillow lava that Jonathan is so excited about.
Yeah, so that tube is no more, but just basically past where the vent for that little guy was the actual breakout from. Yeah, that was just an amazing structure.
Yeah, very unique. And so again, yeah, that was Christopher Ma just joined us from the Smithsonian Institution, the National Museum of Natural History. And here's our first one of these sponges here. Maybe we can take a quick look at that large sponge there.
Feeling any tether heat?
So we see several these primnoid corals
Let me know if you do.
to our left, and then there's a bubblegum coral. That's that red one that we see on the rock in the background, and then there's also a pink coral or a corallium to our left. And yeah, this is the first time we've seen these this massive sponge here on this dive.
Hi, Daniel. It's Chris.
Hey, Chris.
So,
Are you confused to what?
another new one for me. I was originally going to go to Saco Calyx, but I'm not so sure unless it's a really, really old one. It's all twisted and gnarled.
Yeah.
But it's pretty interesting, really nice shot.
Lasers off.
Yeah, I was confused by the stock. I mean, there's several of these morphologies, but when they're not on the stock, and
Yeah, it looks like a lettuce sponge on a stalk, which is weird, but it's probably in the genus Saco Calyx. It's in the subfamily Bolosomini for sure. I think it's a soft sponge.
yeah. Excellent. Thank you, Chris. And you could see before there was a crinoid living on it.
To get an edge to get some contrast.
And then it gives you a nice view of the
Okay.
the porous
But maybe where that crinoid is. With
Sure.
the porous surface of these sponges.
Sounds good. What's that?
Nice.
Okay, I'll hold there.
Yeah, beautiful imagery, and yeah, again, highlighting this time after time is that these sponges and corals are important habitat forming species. They provide habitat for a whole suite of other invertebrates that live in and around these animals, and that's why on this expedition and on this series of expeditions we're targeting some of these environments where we think we're going to find higher abundances of corals or sponges, because as soon as you find those, you'll find all kinds of other associates.
All right, happy with that. Watch lead, anything else to see on this sponge or these corals?
No, that's excellent. Are we stopped or are we on a move?
We are stopped. We are about to move after this shot.
Chris, is there like a big value of getting this? Yeah, I've never seen it. Chris, still there? Yeah, we're having a little bit of trouble with our teleconference line.
Hey, Jonathan. Chris Kelly just stepped out of the room. I just think we probably want to second this.
Yeah, let's. You said move on, right? Yeah, I did not hear him as excited as I've heard him about other sponges.
He can't be too excited if he stepped out of the room.
Yeah, so let's move on.
Okay.
Before we leave, after you're done imaging the sponge, there is a whip coral to the left that there's a request in the chat room to image.
Copy that. Thank you. Yeah. So that wire coral there next to the red.
Sure.
And that's another one of these beautiful views. Is some of these boulders provide prime habitat for corals. So just again, it's just a local high, and then as soon as these things get a little bit off, currents are kind of accelerated around structures like that, and then it provides habitat for all kinds of organisms. And beautiful view here, different colors. So those red, the red, that's a bubblegum coral.
Centered up.
And this is a wire coral, and you can see at the very distal portion of that, it doesn't have any more polyps, so that's the skeleton is exposed. And so this is a unbranched bamboo coral, and might have been something feeding on it on the top. We talked about these sea stars earlier, that they are common predators. Yeah, and then maybe pan up to the top.
Sure. Just shift it a little.
And
Coming partial.
Thank you.
Hi, Daniel. It's Andrea again. I was just sitting here talking to Leslie.
That's good.
This bamboo coral we have here, and he's calling it part of clay sea. But we were just talking about how the sclerites, or those scale-like structures that we see in octocorals are very different from primnoid. And you can see the really long, skinny, needle-shaped sclerites protruding from all of the coral polyps. Really, again, fascinating, great imagery.
Yeah, I couldn't agree with you more, Andrea. And thanks for sharing that. And and you could also see
pretty anyway.
The skeleton there, the exposed skeleton of this unbranched bamboo coral. And bamboo corals, that common name comes from this kind of very unique skeletal arrangement, in that the skeleton has these alternating long white portions and then skinny black portions. Those black portions, the nodes, those are made out of protein, and then the internodes, those white ones, are made out of calcium carbonate. And then but again, this is a very beautiful. Yeah, let's maybe take a quick look at to the bottom of the screen.
The crinoid?
Yep. That's a new one for.
Reading my mind, Copilot. Can you get lasers off too?
So crinoid, but again, this rock again, providing a nice habitat for several different organisms. And the interesting thing is, all of these organisms are suspension feeders. Even this crinoid, so they use their arms there to catch particles that are in the water column. That's the same thing the corals would do with their polyps, is that in all of these organisms, they will have mucus secreted on their arms or polyps, and that way they can catch particles. All of these organisms are found in places where you have dependable currents that would provide them with their nutrients. And this rock here, is a nice habitat feature. There's several primnoid corals. There's a pink coral also to our left, and a bubblegum coral. So a nice little increase in abundance and diversity in this local high. So that's excellent. Yeah, so maybe we can push on here.
Okay.
I'll get a quick establishing.
Sure.
Organism.
What heading are y'all liking best?
This crinoid. There's among the crinoids, there's both stocked and unstocked forms.
Let's just do a quick zoom. There's a little spot lobster on the left.
Yeah, I was wondering about that too.
Yeah.
Oh, it's a little.
That's a ophiuroid.
Yeah.
And those are that ophiuroid on that sponge. That's going to be in the subgroup or family, the Ophiucantidae. So on the these ophiuroids or brittle stars, there's some that have, you see those kind of like hairs on the arms. So when they have those long hairs, that's the family, the Ophiucantidae. And that distinguishes them from the family, the Ophiuridae, which when they don't have those kind of projections.
Okay, whenever you're done. Thank you.
Ready to go. Thank you.
Cool rock.
Yeah, nice little.
Yeah, beautiful view and nice with the dark background. Depth is 1,150 meters.
Northward?
Oxygen, dissolved oxygen right now picked up by the sensors on the ROV is 1.05 milligrams per liter.
Of 30 meters, that's 30 meters.
The temperature right here is at 3.5 degrees Celsius.
2 knots.
And yeah, temperature is one of these things that kind of uniformly cold in the deep, but oxygen, there's this narrow band here in Hawaii around 700 to 1,000 meters where you have lower oxygen levels.
Your lowest swing arms are in the...
that same layer is found throughout the world. It depends on depth.
Did it work to bring it down for that?
It depends on location, so it can be as shallow as 400 meters. But typically in most places in the world you see this layer with lower oxygen concentration at around 400 to 1,000 meters. There's another one of these Seikocalyx sponges, as well as another one of these sea stars. Looks like the same thing that tentative Mediaster until Chris Ma finishes his current description that he's working on. And that's also another common theme, is that a lot of these organisms, we don't yet have the names. There's a lot of research that's happening on them. So the names are changing or still in development. In many cases, we don't have names for these organisms just yet.
Yeah.
Okay, video, let's do a quick zoom. Just floating here.
Oh, I think that is different. I take that back.
Oh, yeah.
Yeah, I would agree. This looks different than what we have seen so far today.
Yeah.
I don't know if you all have seen this on
Similar.
I think that's like Henricia, and I think Chris is probably watching, but yeah, it looks like Henricia.
Are we good with that watch lead, or do we want a steady shot?
No, that's quite common. Yeah, thank you.
Are they going to do the ship move as they rotate their heading?
Yeah, Tina agrees. So there's that Henricia pauperima is a common one, and yeah, it has a very unique kind of texture of the skin.
I just was wondering if we were in hold.
And yeah, it's a nice little aggregation here of more of these primnoid corals, Narella dichotoma, which seems to be the main.
Okay, video snap zoom on this black coral.
Hey Dan, this is Chris again.
Hey Chris.
First and then do the rotation.
Hey, yes, that is indeed Henricia, a globally distributed species that all look alike. They're terrifying, and I'm pretty happy that you just sort of left it there for now.
Excellent.
They're probably feeding whatever small material, organic material is on the rock. Some of them feed on sponges.
Oh, I was talking to Copilot. There's a huge
They occur very commonly in cold water habitats.
No one's talking about the sea pen.
It could
Let's
be
go
that
wide
it's
and we'll
the same
get
one
this
as the one that's in the
sponge
Hurl
before it
Guide
gets
and
us.
seen in shallower Hawaiian waters, but probably not, because oftentimes these species are differentiated by depth. And looking at it closely would be the only way to even guess at whether or not it was separate. And there are some species that are technically one species, but molecular data suggests there's more to it than that. So it's a complicated animal in spite of looking so simple. So
the ship move started. So
happy
couple
to
minutes
sort of
before
leave that to
push ahead.
other people for the moment.
Excellent, thank you.
Thank you for taking a quick look at it.
Thanks, Chris, and there's one of these massive sponges, and that's also on a stalk, which is quite interesting. Maybe one of these massive Sacocalyx, and we also passed right after the sea star by a rock pen, or there's a sea pens, and that's in the genus Anthoptylum. And this is quite spectacular. This massive stalked sponge.
Okay, we've got those enough. I'll turn
And just to give you a little bit of a feel, so that's more than a meter in diameter, that top portion. So that's a big animal.
Got different sea star below it too.
A little pudgy one. Okay, video. I'm floating here. We'll do a zoom on the sponge just to record it, and then I'll move down to the sea star.
And Scott France from the University of Louisiana is pointing out it's very interesting to see such a massive animal in such a narrow, small
walk across the bottom, like to port.
Stalk.
Yeah, just walk across the sponge at partial. We can kind of, might be the best way to see something so big.
Yeah.
Probably also say something, he mentioned it says something about buoyancy, but it probably also says something about the current and that they're not too vicious here. Otherwise, they would probably be toppled over.
It's amazing.
Yeah, it's big.
Dad, it could be useful to have the lasers on if they aren't on. I can't tell, the video's dropping out a little bit, but we could get a size scale with that.
Yeah, they were on when we had the shot of the entire colony, Bruce. Yeah, so that's where I got that estimate.
Okay.
So this is over a meter in diameter. This this top portion of the sponge.
Lasers back on.
Yeah, go for it.
Beautiful animal, yeah, absolutely spectacular.
Let's capture the whole thing again with lasers.
Roger.
And just absolutely beautiful scene here with the dark background.
If you're done with cereals, can we zoom out?
And a lot of these organisms taking on very, very peculiar shapes here in the deep sea, and this is something that a lot of people always comment on, is that it looks like it's on another planet. But one thing I always like to point out that this is actually the more common things to see is that the vast majority of our planet is found in the deep sea. The oceans are a lot deeper than the land is high. The vast majority of our planet is
video. We'll zoom on
underwater, and the vast majority of our planet is in very deep water. Here we have another sea star.
Maybe that looks like the one we saw.
I'm sure Chris is still watching attentively.
Daniel, it looks like there's another scale worm on that sea star as well.
Yeah, exactly. Good eye. So those are the pollinoid polychaete worms, and when you zoom in, you can see that they have tiny scales. And with the quality of this video, so apparently that's a common association here.
For the clipper, something just went by.
And pollinoid scale worms are commonly found in associations with all kinds of animals. They live on anemones, on black corals, on other Gorgonian corals, on sea stars. And
once I hold it, I'll move on.
Great.
You happy with that?
Yeah, excellent.
Thank you, Bridge.
So beautiful view of this sponge, of this massive sponge. Depth right now is 1,140 meters. So we'll
get over this
try to cover some ground here. So we are approximately 2 hours and a half into this 9-hour dive. There's a nice Prosingit sea star that, and that's going to be in the genus Brasinga.
I start shooting up.
And that's very conspicuous for those sea stars that have very, very long arms.
So we
and the central portion is very short.
We're turning.
Oh, another one of these.
Yeah, but
Or then
the
ship move.
The bearing of the ship move.
So another one of those enormous stalk sponges. And Chris had suggested that sponge there might be a Seiko calyx. We had only seen them in the very small sizes, like the one to the left there. But apparently here several of them are bigger than we've ever seen for that group.
Okay.
And yeah, they start growing a lot more complex when they're small, like the one that's on the left side of the screen right now. They're perfectly round, and perhaps as they get bigger, they get more amorphous, so they take on different shapes, and it's not perfectly circular anymore.
So it looks like the slope is more to the east now.
On your sonar? Yeah.
Yeah.
That should be right, because we're starting to head off to the west of the ridge and high pack, so we might have to adjust to...
Yeah, something like 2.5.
Yeah, I'm looking at some rocks right here that might be the center.
Yeah, we got plenty of space now. There was like a little cliff, and then it leveled off a little bit.
Yeah.
So coming into view right now are some pillow lavas, actually. So this is the other...
Ready for another move?
Other type of lava flow morphology that I was talking about earlier in the dive. And if you're just now joining us, we're currently on an unnamed seamount that's just east of Salmon Bank, about 1,250 miles away from Honolulu. We just mapped this seamount for the very first time using our sonar mapping and generated high-resolution images of it, and now we are currently diving on it for also the very first time. We have already collected one geological sample at the very beginning of the dive. The geological sample is going to be crucial in learning a whole lot about this seamount, including its age, its chemical composition, and its history as the volcano evolved. So these more rounded shapes to the rocks that we're looking at is actually the primary structure being concealed by the manganese crust that's growing on top of it. So these larger, more rounded shapes to the rocks we're looking at right now are what I just called a pillow lava. And these are one of two common morphologies that lava flows take on as they erupt onto the surface of the volcano underwater. Looks like there's one of them that actually kind of looks like it's been broken in half and dissected. You sort of get an idea what they look like on the inside.
Kind of top screen, yeah, hold there. That's good. Coming up.
Yeah, it's a nice zoom. Yeah, so that smaller globular structure or texture that's on top of the rocks that we're looking at is a manganese crust that is a precipitate that starts to grow on top of the surfaces of rocks. The texture that we're looking at, it's one of my favorite words to say. I keep on saying it's got a botryoidal texture. Botryoidal comes from the Greek word of bunches of grapes, and you can kind of see the relationship and why we call it that. Getting a nice good zoom in on the botryoidal texture of the manganese crust. Very peculiar type of texture. So we're currently at a depth of 1,124 meters. We got to the bottom at approximately 1,261 meters. So trying to make our way to the north up this ridge where we will be on top of one of the main volcanic rift zone ridges. The rift zone ridges of these seamounts and volcanoes, the locus point of many of the eruptions as the volcano is growing. The material that is upwelling in the mantle deep beneath the Earth's crust starts to melt. That turns into magma. The magma then rises into a shallow magma chamber, and the magma chamber will end up becoming too filled up and pressurized with the magma. When it becomes too pressurized, the magma actually cracks the crust along these rift zone ridges and pushes the two sides of the volcano's ridge apart. This is where we get the rift part of the rift zone ridge. So any kind of geological formation which has two different bodies of rock moving away from each other, we term that a rift. So that's where we get the rift zone ridge terminology for these types of features. As the magma intrudes into the subsurface and pushes the ridge flanks apart, the magma then either stays under the subsurface where it cools slowly and turns into what we call a dike, or it will actually break the surface where it starts to erupt. And when it erupts, it forms these pillow lavas on the surface, either pillow lavas or sheet flows. And sheet flows are the more tabular structures that we actually saw a little bit earlier on the dive.
Looks like we're halfway through a move.
Thanks, Dev.
Pilot video, comm check.
Hey there, video.
Hey, all your cameras are full wide.
Thank you.
No.
Sorry to backtrack, I just saw something. There are 2 sea stars, I think, on this. There's at least one.
A little bit of time here, but once you're done, you're going to have to go
Come on
up
in.
slope.
Coming in. Aye.
Hey, Jonathan, there's something on the left as well. It's purple. If we could zoom in on that. Oh, it's a crinoid. I see it now.
Copy that, Andrea. Would you like a zoom?
I think we've have several zooms of that. I wasn't sure what it was from afar.
Okay, yeah, you can kind of see its arms in the left portion of the video, but it's a nice zoom of the sponge skeleton sea star.
You're going to have to go pretty soon.
Okay.
And coming out.
Then there was a shrimp below the sea star, kind of on the other side of the skeleton.
Yeah, copy that, Bruce.
See the slope not too far ahead.
Hey, Dan.
Starting to look down.
Hey, Chris Ma. This is Jonathan. So,
hey,
yeah.
Looking at your starfish.
Yeah, how's it going?
So this is another Goniasterid. It's similar. It's the same one I think you saw just at the last stop at the base of the sponge. It looks like sort of. It's a Goniasterid. It looks like something called Ceramaster, but that's a name that gets applied to a lot of things. It's also very possible that there's another family that resembles Goniasterids called Odontosterids, and you can only tell from an examination of the underside. And they feed on sponges, but goniastrids certainly feed on sponges also. So, and we don't really know a lot about what a lot of goniastrids feed on. So the fact that it's on top of that animal, probably, digesting the syncytium or whatever is edible on a glass sponge is novel. So I'll have to sort of look at the picture more and see if it corresponds to, I think there's one in this area called Ceramester Bower's eye, what used to be called Nereodaster. So I'll have to look into that. But it's, like I said, everything here is new to me. It's fascinating.
I'm ready to call that.
And it has a commensal scale worm on it too, which is, sort of a big double whammy today. So exciting. I'm really kind of glad to see these. These were the animals that I worked on for my dissertation.
Great, awesome. Thank you, Chris. So that was Chris Ma from the.
Thank you. I'll talk to you later.
From the Smithsonian National Museum of Natural History. He specializes in these sea stars and is always very willing and waiting to call in and give us some very interesting factoids on each one of these species identifications and some of their natural history. So thank you, Chris.
And it looks like we have another really large sponge here. I think that's the 4th observation today.
Yeah, copy that, Andre. You want to zoom? I'm sure Chris would appreciate a zoom.
Sure thing. Settle in on it here.
Yeah, sure. If you're moving along, you don't have to stop, but it's not zoom.
Come on in. Video zoom. When you're ready. Yep. Okay, I will. All right, I will enunciate well and speak clearly and loudly. Roger that. Let me know if you see anything in there that you want to take a longer look at. Yeah, this one seems... Sorry, I was just starting a little pirouette around. I had to fight it. There's something pink back in there. You can tell there's something living back in there a little ways.
So Nav, as soon as he's done looking at this, we'll call in that move.
Copy that.
Yeah, pilot, I think that's good for now. So if you guys want to go ahead and get on the move.
Why don't you take a snap in on this part? I think this will be cool against the black.
You're thinking,
oh, yeah, definitely.
30 meters, 015.
Yeah,
pilot?
just see how...
Okay, cool.
Yeah, okay. Oh, that's fine.
We'll wait.
I'm trying to get the internal structure a little better.
Imaging.
Yeah, so that's a beautiful zoom illustrating the porosity of these animals. They, being filter feeders, rely on the fact that their tissue is very porous, enabling fluid and current flow to transport nutrients through their tissue. They have specialized cells that actually encourage increased flow and current through their tissue.
Yeah. Is this full zoom? Okay. Alright, we can hold this as long as you like, video, and then I can run off. Continue the moves.
Hey, Jonathan, we're good here with the close-up of the sponge,
really
so whenever you're ready to move on.
Copy that, Andrea. Yeah, we're just trying to get this nice.
On the rest of the sponge.
Yeah, we're just trying to get a nice zoom with the black background and the translucent tissue. It's making for some good imagery. So after we get this zoom, we're gonna get on the move again and see what else there is to see. Thank you.
Copy that. Thank you.
Thanks a lot.
All right, I'm going to call the move in, boys.
Yeah, go for it.
Okay.
Thank you.
Bridge, this is Nav. I'd like to request a jet move.
Just stepped out here for a second to grab lunch and
at a speed of 0.2 knots.
Roger that, co-pilot.
Current depth is 1,110 meters.
Good copy, Bridge. Thanks.
The dissolved oxygen content is still right around 1 milligram per liter.
It's hard to judge how high I am with this. Video kind of dark. Let me look down. I just thought I was a lot higher up because the video was. This monitor throws me off a little bit, but I've got my minute cam, so I'm all right.
I just passed a couple of these mushroom corals. Those red, also very distinctive.
Yeah. Okay. Do what you gotta do.
Thank you, Bridge.
So again, to give everyone a heads up, today's dive is going to be a late recovery. So we just have just under 6 hours remaining of bottom time. We're at a depth of 1,100 meters. We've ascended about 200 meters up this slope, and we want to make it to the top, which is at around 700 meters. Floating in the water column, probably another one of those Aerostage shrimp.
If you want any snap zooms, let us know. Video or watch lead with videos. Just trying to sort out a little.
Roger that.
We're operational over here. We think we're not sure.
Okay.
That sponge in the background looks slightly different. Maybe we can take a quick snap zoom.
And it does look like there's a little fly trap or something on it.
Yeah. So that's an anemone on top of the sponge. Also common.
Thanks.
In particular with that common name of those are the
video.
Venus flytrap anemones.
Roger, coming in on the sponge.
So this is a different species. So this is the species Pharia near Oca erecta. There's a squat lobster on the top, and then there's also that Venus flytrap anemone that's going to be in the genus Actinoscyphia. Also in the background, there's a solitary cup coral to the bottom of that sponge. Yeah. And yeah, a couple ophiuroids, albedo stars growing on this. So yeah, nice little assemblage here.
Wherever you need video.
So I think that squat lobster looks like Gastroptychus.
Let me just come in for
closest Gastroptychus iaspis.
Maybe the boundary between the two there on the left a little bit.
Between the two animals here.
Yeah.
beautiful view here of this Venus flytrap anemone.
That'll. I guess you're fine.
Do they want the squat at all, pilot?
Hi, Daniel, Sandra. I was just thinking it's a nice shot of a different phyla. We have the Nidaria, Peripera, Kynodromata, and the Arthropoda.
Yeah, excellent. Yeah, good.
All on one little area here.
Good point. Yeah. Yeah, and again, the point we make time after time is that how important these habitat-forming species are. Once you do see these massive corals, sponges, you see all kinds of other animals on them.
How are we doing on that move, Nav?
I think we're stopped, aren't we?
No, we got maybe 10 meters left, not even.
Let's
all right,
keep it
coming out a little bit.
Roger that video.
And Tina even says she's
that was a 30-meter move.
Spotted some copepods. Excellent, thank you.
Yeah, of course. Here's a Roland zoom. Let's get this disc. He likes these.
Yeah.
They're fun to get once in a while.
Yeah, so that's going to be Ophiuroid, and it's going to be in the family Ophiocantidae.
Second or two, and we can pick up. There's a rock off in the distance. I might have some fun stuff growing on it.
Coming wide now.
Yeah, and
see if there's anything of interest on this rock, and if not, we'll extend the ship move.
You can extend it. I don't think.
Okay.
Excellent.
Rods down there on the
so
bottom
I
left.
did note here the ID that Chris and Mary provided. And yeah, I just pulled up this photo here of the genus Pseudomonida, and it does look
if you
very similar.
Underway, you're directly over some sort of a straight rod.
Yeah, we are moving along, and we're calling in another one.
All right. So
And yeah, interesting that just we started seeing now a few more of these Pharia neuroca erecta sponges, these phariid sponges. The primnoids are still the same things the Narella. It looks much like Narella dichotoma, still the same, but yeah, we're starting to see other sponges appear.
Have we seen these yet? This coral?
Yeah.
Bridge, this is Nav?
Yeah, we have.
Okay. Continuing on.
Yeah, I'd like to
and Andrea is also pointing out that we are seeing fewer corals here. Now the habitat also appears to have changed.
I copy 010.
The ridge isn't as well defined here as the place we were earlier.
30 meters, sorry.
Good copy, Bridge.
You lined up on.
There's another one of these large Pharia neuroca erecta sponges that's now dead to the bottom.
Lack of anything else.
And there's a
shot. Come on in, video.
Coming in.
If I can get it, that'll be a cool shot.
Yeah, beautiful shot of this shrimp.
I'm going to,
thank you, Bridge.
Hold at this. Don't want to push our luck.
I'm just going to go ahead and update our position here.
From us, yeah. Must not like us.
You let him go. Just let all the antenna out of frame too, or whatever they are.
Great.
Great. Thank you very much, pilot. Nice work.
We see these glass sponge before?
Maybe. Yeah, but yeah, yeah, it is a glass sponge. So yeah, let's take a quick snap zoom maybe.